Today we are going to look at Chapter 9, Pulmonary System and Exercise. Objectives. Diagram the ventilatory system and show the glottis, larynx, trachea, bronchi, bronchioles, alveoli. Describe the dynamics of inspiration, expiration during rest and exercise. Describe the Valsalva maneuver and its physiological consequences. Define minute ventilation, alveolar minute ventilation, ventilation perfusion ratio, and anatomic and physiological dead spaces. Explain the Bohr effect and its benefit during physical activity. List and quantify three means of carbon dioxide transport in the blood. Identify major factors that regulate pulmonary ventilation during rest and exercise. Describe how hyperventilation extends breath holding time but can have dangerous consequences in sport diving. Graph relationships among the pulmonary ventilation, blood lactate concentrations, and oxygen uptake during incremental exercise. Indicate the demarcation points for the lactate threshold and onset of blood lactate accumulation. Explain what triggers exercise-induced asthma and identify factors that affect its severity. Pulmonary functions. They supply oxygen required in metabolism, eliminate carbon dioxide produced in metabolism, regulate hydrogen ion concentration, H+, to maintain acid-base balance. The anatomy of ventilation. This is on page 252. It shows the nose and mouth, trachea, bronchi, bronchioles, and alveoli. So here's the picture. You have the nasal passage, you have the oral cavity. So you have the oral cavity, pharynx, larynx, cartilage. Then you have your when the breaths come in, they go into the lungs and they end up, it's like a tree-based thing, which is the bronchi, then the bronchioles, and the alveoli, which is even smaller, which are like little sacs where you have the capillaries and pulmonary arterioles that where carbon dioxide and oxygen, that gas exchange takes place. So yeah, you have oxygenated blood that's able to go out to the tissues, and then you have the CO2 that has to be expelled out of from your breath to get rid of that byproduct from the cells functioning. Inspiration diagram contracts, flattens out, moves downward. Air in the lungs expands, reducing its pressure. Pressure differential between the lungs, the ambient air sucks air in through the nose and mouth and inflates the lungs. Inspiration concludes when thoracic cavity expansion ceases and intrapulmonic pressure increases to equate or equal atmospheric pressure. Predominantly passive process. Air moves out of the lungs from the recoil of stretched lung tissue and relaxation of the inspiratory muscles. Sternum and ribs swing down while the diaphragm moves toward the thoracic cavity. So when the sternum and ribs swing down, okay, expiration, the diaphragm moves up toward the thoracic cavity. These movements decrease chest cavity volume and compress alveolar gas, forces it out through the respiratory tract to the atmosphere. The static lung volumes, you have tidal volume, which is air moved during either inspiratory or expiratory phase of each breathing cycle. You have inspiratory reserve volume, And there's a chart of this on 255, which is an additional volume of, so going back to tidal volume, it ranges back to 0.4 and 1 liter of air per breath. Inspiratory volume is an additional volume of 2.5 to 3.5 liters above the tidal volume. That is the reserve for inhalation. Expiratory reserve volume is after normal exhalation, an additional volume that can be exhaled. So this is additional volume can be exhaled after a normal exhalation. Okay, 1 to 1.5 liters per man, 10 to 20% for women. So IRV is after a breath and inhalation, how much you can still breathe in. Expiratory reserve volume is after exhalation after a normal breath cycle, how much can still be exhaled. Forced vital capacity is total air volume moved in one breath from a full inspiration to max expiration. Varies with body size, body position when measuring. Residual lung volume following a max exhalation volume of air that remains that cannot be exhaled after you try to basically, after your ERV, your expiratory reserve volume, the residual lung volume is what's left that can't be exhaled, still in your lungs, which is 1.2 to 1.6 liters per man, 1 to 1.2 liters for women. And this just shows what that is. So you have a tidal volume, your inspiratory reserve volume, expiratory reserve volume, forced vital capacity, your functional residual capacity, your TLC, which is your total lung capacity. Ooh. Okay, and then your RV, which is your reserve volume. Okay. All right, dynamic lung volumes. You have dynamic measures of pulmonary ventilation depend on max stroke volume of lungs, speed of movement of air. So this is kind of like the pulmonary ventilation. It's kind of like how the heart, the cardiac output, where you have the stroke volume. So how much is in one breath, and then how many breaths are you taking? Okay, that's what this is. Okay, so you have FEV, percentage of FCV expelled in 1 second. Okay, so forced vital capacity, which is max volume expired after max inspiration in 1 second. And then you have FEV, FEV divided by the FVC reflects the expiratory power and overall resistance to air movement in the lungs, averages 85%. All right. Max voluntary ventilation requires rapid deep breathing for 15 seconds. That's extrapolated to the volume breathed for 1 minute. Ranges between 140 and 1 liter per man, 80 to 120 for women. And there's on page 9.1, on page 257, table 9.1, it looks at the shallow breathing, normal breathing, and deep breathing, and how minute ventilation can still be the same if the shallow breathing equals a high breath rate. The deep breathing could be a few deep breaths, but then the rate doesn't have to be as great, obviously. Minute ventilation is breathing rate times tidal volume, and this is like cardiac output. You have the stroke volume times the heart rate. Okay, it's very similar. It's analogous to that. So you have the breathing rate times tidal volume, which is how much in a breath, times the breathing rate. Okay. So you have 12 breaths at 0.5 liters equals 6 liters per minute. Alveolar ventilation, the portion of minute ventilation that mixes with the air in the alveolar chambers, determines gaseous concentrations at the alveolar capillary membrane. Okay, this is basically the portion of minute ventilation that mixes with the air in the alveolar chambers, where it's like the gas exchange. Okay, is really what it's just talking about, the gaseous concentrations at the alveolar capillary membrane area. Anatomical dead space, the air that fills the nose, mouth, and trachea and other non-diffusible conductive portions of the respiratory tract at any time. Okay, so right now there's anatomical dead space as I'm talking, where there's nose, mouth, and trachea and other non-diffusible conducting portions that I'm not breathing in or out. Okay, 150 to 200 milliliters, about 30% of resting tidal volume in healthy people. Physiological dead space, the portion of alveolar volume with poor tissue regional perfusion or inadequate ventilation. Physiological dead space can increase to 50% of resting tidal volume because of inadequate perfusion during hemorrhage or blockage of the pulmonary circulation for embolism or blood clot. Okay, and then inadequate alveolar ventilation and chronic pulmonary disease like COPD. Okay, an embolism, by the way, is a obstruction of an artery, typically like by a clot or blood, a clot of blood or an air bubble. That's here about pulmonary embolism. Physiological dead space is the yellow in the middle there, and that talks about tidal volume, why the tidal volume possibly, what part constitutes that physiological dead space. Okay, disruptions in normal breathing patterns. You have dyspnea, which is shortness of breath or subjective distress in breathing. Hyperventilation is an increase in pulmonary ventilation that exceeds the oxygen need for metabolism. And another condition that's sometimes talked about is hypoxia, which is the absence of enough oxygen to the tissues to sustain bodily functions. Gas concentration, the amount of gas given in a given volume determined by the product of the gas's partial pressure and solubility. Gas pressure, the force exerted by gas molecules against the surfaces they encounter. Partial pressure is the percentage concentration times total pressure of gas mixture. Ambient air, it's percent oxygen, 159 millimeters of mercury, and then percent carbon dioxide is 0.2 millimeters of mercury, and then nitrogen is 600 millimeters of mercury. So you see that the ambient air is like about 79% nitrogen. Okay, tracheal air, percent oxygen is 149 millimeters of mercury, percent carbon dioxide stays the same. Alveolar air, percent oxygen 103 millimeters of mercury, percent carbon dioxide is 39 millimeters of mercury. Henry's law is the amount of gas dissolved in a fluid depends on pressure differential between gas above the fluid and below, and the pressure differential between the gas above the fluid and dissolved in it, and the solubility of the gas in the fluid. And this is just this here really talks about diffusion. Okay, solution of oxygen and water. So you have diffusion where you have a high concentration to low. That's why above the water you see 160 millimeters of mercury. It's pressure of oxygen. It's going in, diffusing into the water. Then you have B. You have 160 outside the water, and then the fluid. Or yeah, it's water in this example. And then and so you still have you still have diffusion that's going from outside to inside the water. More so, you do have a little bit of oxygen going out because there is some pressure. But then there's it's homeostasis where you have 160 and 160, so it's there's a balance. Gas exchange in the body, the exchange of gases between lungs and blood, and their movement at the tissue level takes place passively by diffusion. In the lungs, first step in oxygen transport involves transfer of oxygen from alveoli to the blood. In the tissues, oxygen leaves capillary blood and flows toward metabolizing cells, or carbon dioxide flows from the cell into the blood. What this means is basically oxygen is leaving capillary blood and it's going towards the cells, okay? So cells can do work. Carbon dioxide is flowing from the cells to the blood so that it can be taken back to the lungs and then expired so we can get rid of the carbon dioxide, okay? Gas exchange continue. This is on 263, and it just shows inspired air comes into the trachea. What's the percent oxygen? 149 millimeters mercury. Makes sense because you're breathing in air, and then you have 0.3 millimeters of mercury, the pressure there of CO2, carbon dioxide. Arterial blood is 100 millimeters of mercury, 40 millimeters of mercury, CO2. Okay, and it's going through that pulmonary capillary, and then you're going to out toward the tissues. You are still having a high amount of oxygen toward the tissues. Skeletal muscle uses the oxygen, okay, and then after that, you see what happens. It changes, and you have then a higher pressure of CO2, carbon dioxide, 46, and then lower at 40 millimeters of mercury for oxygen. Going to the venous blood, back to the pulmonary capillary, the trachea breathes out that carbon dioxide. And where that happens at is in the alveoli, and the alveoli is the sacs in the lungs. Oxygen transport in the blood and physical solution dissolved in the fluid portion of the blood establishes the percent oxygen in blood and tissues combined with hemoglobin in loose combination with iron, protein, hemoglobin molecule. Red blood cell increases the oxygen's carrying capacity. So hemoglobin carries oxygen. It's an iron-based. Hemoglobin carries oxygen. Okay, it's in the blood, and it consists of protein globin, composed of 4 subunit polypeptide chain. Each polypeptide contains a single heme group or iron group with its single iron atom that acts as an iron magnet. So this hemoglobin is an iron atom that carries oxygen. Okay. So it increases blood's oxygen carrying capacity to 60 to 75 times above that have normally dissolved in plasma. Each 100 milliliter of blood contains approximately 15 to 16 grams of hemoglobin in men. 5 to 10% less for women, or 14 grams per 100 milliliter of blood. This is on page 266. It just talks about how it's an oxyhemoglobin dissociation curve. The dotted line represents myoglobin, which is the iron and oxygen binding protein in muscle, and then hemoglobin is the solid yellow line. It just shows that around 60 millimeters of mercury, a sharp decrease occurs in how much oxygen combines with hemoglobin. So oxygen dissociates from hemoglobin at around 60 millimeters of mercury. It doesn't happen and that's where you get the S curve. Okay, in the small, you have the effects of temperature, effects of acidity. Okay, basically, decrease in an increase in acidity causes oxygen to dissociate. So then you're going, increase in acidity is going back from towards 0 from 7 as a pH. And then also a decrease in temperature, um, excuse me, an increase in temperature increase causes the oxyhemoglobin dissociation curve to shift downward to the right to reflect enhanced unloading. So decrease in temperature, an increase in temperature also causes oxygen to dissociate from uh, hemoglobin. This is called the Bohr effect, and this results in alterations from uh, hemoglobin's molecular structure from blood chemistry and temperature changes. It's named from Danish physician and physiologist Christian Bohr. Bohr effect becomes particularly important in vigorous activities because increased metabolic heat and acidity in active tissues augments oxygen release. And B, the chart at the bottom, term oxygen transport cascade characterizes partial pressures. Oxygen moves from ambient air at sea level to the mitochondria of maximally active muscle tissue. Okay, the partial pressure declines. Carbon dioxide transport in blood, physical solutions in plasma, 7 to 10% establishes the percent carbon dioxide of the blood. Loose combination with hemoglobin, 20%, combined with water as bicarbonate. And bicarbonate is then gets changes into carbon dioxide and water, okay, to be expelled through breath and then also sweat. And that's oxygenation of macronutrients as well. Carbon dioxide transport in blood, you have CO2 dissolved in plasma, okay, you have the red blood cell that comes along, so then you have the carbon dioxide is carried by the RBC, and then you see that so it's CO2 is combined with water as bicarbonate, and then you have so you have physically dissolved in blood plasma. You have chemically for B you have chemically bound to hemoglobin, and then combined with water as bicarbonate is what you see with C there. Ventilatory control during rest, neural factors. Respiratory cycle comes from inherent automatic activity in inspired neurons. So at rest, it's automatic, right? I mean, you don't have to think about breathing. Exhalation begins by passive recoil of stretched lung tissue and raised ribs when the inspiratory muscles relax. Activation of expiratory neurons and associated muscles that further facilitate expiration. As expiration proceeds, the inspiratory center is released once again from inhibition and progressively becomes more active. Neural factors continued to ventilatory muscles. You have the respiratory center in the medulla that deals with the normal respiratory cycle. Okay, the inspiratory neurons, basically the medulla. The normal respiratory cycle comes from inherent automatic inspiratory neuron activity whose cell bodies resides in the medial medulla. Lungs inflate because the neurons activate the diaphragm and the intercostal muscles. The inspiratory neurons cease firing for two sources. their own self-limiting and inhibitory influence from the medulla's expiratory neurons. Inflation, of lung tissue stimulates bronchial stretch receptors that inhibit inspiration and stimulate expiration. So you have temperature receptors in the lungs, proprioceptors in the joints and muscles, chemical state of blood and medulla, peripheral chemoreceptors, motor cortex, and subcortical regions. Okay, which cause this automatic process to take place, this ventilatory control during rest. The humoral factors, which is the chemical state of blood that largely regulates pulmonary ventilation at rest. Variations in arterial PO, the percent oxygen, percent carbon dioxide, acidity, and temperature activate sensitive neural units in the medulla and arterial system to adjust ventilation and maintain arterial blood chemistry with narrow limits. Chemoreceptors are their structures that stimulate ventilation in response to increased carbon dioxide, temperature, and acidity. A decrease in oxygen and blood pressure, and perhaps a decline in circulatory potassium. Carbon dioxide pressure in arterial plasma provides the most important respiratory stimulus at rest. Okay. Then you have carotid body, carotid artery, and then aortic bodies. Okay. Ventilatory control during exercise. Chemical factors. You have percent oxygen. So arterial percent oxygen in exercise does not decrease to the point that stimulates ventilation by chemical or chemoreceptor activation. PO2 percent carbon dioxide and hydrogen concentration. Okay, hydrogen ion concentration. Peripheral chemoreceptors provide an early warning and detection to alert against reduced oxygen pressure. These structures also stimulate ventilation in response to the increased carbon dioxide, decrease in blood pressure, temperature, acidity, perhaps circulating potassium. That's what we talked about already. Okay. The term hypoxic threshold refers to the point at which decreasing arterial percent oxygen stimulates ventilation and usually occurs at an arterial percent oxygen between 60 and 70 milligrams of mercury. So you have these specialized neurons located in the aortic arch, aortic bodies, and branching of the carotid arteries in the neck. Chemical stimuli can't fully explain the hypernia during physical activity. Okay. Hyperpnea. You have cortical influence, which is neural outflow. So the neurogenic factors. You have the cortical influence, which is neural outflow from regions of the motor cortex during exercise, and cortical activation in anticipation of exercise to stimulate respiratory neurons in the medulla. So these are neurogenic factors. Okay. So when activity begins, cortical outflow acts in concert with demands of physical effort, abruptly increases ventilation. Peripheral influence. Sensory input from joints, tendons, muscles adjusts ventilations during physical activity. Specific peripheral receptors remain unknown, but experiments involving passive limb movements, electrical muscle stimulation, voluntary physical activity with the muscle's blood flow occluded support the existence of mechanical receptors in peripheral tissues that produce reflex hyperpnea. And hyperpnea, by the way, is increased ventilation. That's what the term means. Integrated regulation, a single factor controls breathing and exercise. Phase 1 ventilation is neurogenic stimuli from the cerebral cortex, and active limbs cause the initial or abrupt increase in breathing when exercise begins. So you see, you have active limbs, things like that cause phase 1 ventilation. All right, and neurogenic stimuli from cerebral cortex. Moving take place, and then ventilation begins relative to that. Okay, to make up for that. Phase 2 ventilation, central command inputs plus medullary control system, neurons and peripheral stimuli from chemoreceptors and mechanicoreceptors contribute to the control of minute ventilation and gradually increasing to a steady level. Phase 3 ventilation, fine tuning of ventilation through peripheral sensory feedback mechanism. So the phase 3 basically central and reflex stimuli from the main byproducts of increased muscle metabolism. Carbon dioxide and hydrogen concentration modulate alveolar gas pressure in this phase. These factors stimulate chemoreceptor group 4 unmyelinated neurons that can communicate with the central nervous system to regulate cardiorespiratory function. So you have integrated regulation. You have oxygen uptake as it goes up. You have minute ventilation that goes up, and what causes that? Neurofactors, uncertainty, like the uncertain factors there. Temperature causes integrated regulation and pH. Ventilation during steady rate exercise. Ventilation equivalent for oxygen is VE divided by VO. Ratio of minute ventilation to oxygen uptake remains relatively constant during steady rate exercise. Ventilatory equivalent for carbon dioxide is VE divided by V CO2, which is carbon dioxide. Ratio of minute ventilation to oxygen produced remains constant during steady rate exercise because pulmonary ventilation eliminates carbon dioxide produced during cellular respiration. Ventilatory threshold, point at which pulmonary ventilation increases disproportionately with oxygen uptake during graded exercise, relates directly to carbon dioxide's increased output from the buffering of lactate that begins to accumulate from anaerobic metabolism. So you have anaerobic metabolism we talked about, which is like the glycolysis. You have ATP system, phosphocreatine. Then you have glycolysis. Oh wow, then lactate's building up. So what happens? You're breathing at a minute ventilation rate that's, steady. Then you have steady lactate accumulating. Then it starts to really accumulate where you have the onset of blood lactate accumulation by either exercise intensity, or it's coming from the lactate system, the anaerobic system, or you have just steady state increase even from the aerobic system. And so then what happens is you have that point of ventilatory threshold where you have to start breathing more quickly. That's called the respiratory compensation. And that's what this shows. It shows that you have, point of lactate threshold, lactate starts going up, start getting this burning in your muscles. And then you have to really, and it may not even happen, you may not get burning until after lactate's starting to already accumulate. But the compensation has to take place to try to get rid of that blood lactate, okay? And that's why breathing increases. Onset of blood lactate accumulation indicated by eventual sharp upswing in pulmonary ventilation related to oxygen uptake during incremental exercise. Implies an imbalance between the rate of blood lactate appearance and disappearance. Occurs between 55-65% of VO2 max in healthy untrained subjects and often equals more than 80% of VO2 in highly trained endurance athletes. So what this is telling you is if you're trained, if you're in better shape, your blood lactate accumulation doesn't happen as quickly because occurring at more than 80% of VO2 max rather than 55 to 65. The point of OBLA often increases with aerobic training without any accompanying increase in VO2 max. Okay, so your VO2 max may not increase, but your OBLA often does with aerobic training. And this just talks about onset of, this is on page 276, just talks about 5 possible underlying factors that relate to detecting lactate threshold for pulmonary gas exchange dynamics during physical activity of progressively increasing intensity. So you have inadequate oxygen delivery or utilization. Then you have, from the anaerobic metabolism, you have increased lactate, the buffering of sodium bicarbonate, things like that. Or you have the delayed steady rate of VO2, okay? That doesn't come, maybe, it possibly doesn't come from the anaerobic metabolism, it comes from steady state, okay, maybe aerobic. Then you have minute ventilation that then non-linearly increases, delayed steady state, steady rate, constant work test, and then respiratory compensation for metabolic acidosis. So that can cause the onset of blood lactate accumulation. It doesn't have to be from the anaerobic system, it can come from the aerobic system of steady state. Work of breathing. Breathing normally requires a relatively small oxygen cost, even during exercise. In respiratory disease, the work breathing becomes excessive. In exercise, alveolar ventilation becomes inadequate. Factors determine the energy requirement: breathing, compliance of lungs and thorax, resistance of airway, smooth flow of air. Buffering. Buffering consists of a weak acid and the salt of that acid. The bicarbonate, phosphate, and protein chemical buffers provide rapid first line of defense against acid-based regulation. Lungs also contribute to pH regulation through change of alveolar ventilation, rapidly alter the free hydrogen concentration in extracellular fluids. The renal tubules act as a body's final defense by secreting hydrogen ions into the urine and reabsorbing bicarbonate. So renal tube is kidney tubules, okay? Do that. And then anaerobic exercise increases demand for buffering and makes pH regulation progressively more difficult. Buffering continued. And this basically just shows you that when your when your pH drops, okay, your blood lactate is going up. Okay, that's the bottom there. And it says that blood pH, okay, when it becomes more acidic, the blood lactate concentration is going up. The top chart is the blood lactate concentration is going up, okay, as the pH drops, becomes more acidic. So as lactate increases, the blood becomes more acidic. Then on the bottom, percent max exercise, you have blood pH is going up. Percent max exercise is going down. Excuse me. Percent max exercise, really, as it goes up, okay, then you have the blood pH that rises, and then your blood lactate rises. Okay, that's the yellow line. The blood lactate rises as the exercise goes up, obviously, because lactate increases relative to percent max exercise. And as that's going up, your blood pH drops because it becomes more acidic. Excuse me.